Greetings from Sweden, where I'm recording this session. I hope to be meeting with you later as you've listened to this. I want to thank the organizers for inviting me to present this, Introducing Nurture Science, a new model with ancient roots. My recording isn't working here. So, Nurture Science is also the name of my business. My company is called Nurture Science for Improved Neonatal Outcomes, as in Nino. Now, nurture and science are an oxymoron. They're almost like contradictory, incongruous words from my training. And nurture comes with three definitions. There's a nurturing into a secure being. Especially in the United States, nurturing is often used as in feeding. And then there's the other nature versus nurture meaning of the word. And my nurture science covers all of these three in a very inclusive way. Now, the ancient roots, in modern parlance, we could call that life history theory. And I want to share a few pictures from the anthropologist's first meeting with some people that had never met any other people ever. And they've lived like this for 80,000 years. They were hunter-gatherers. The women gathered, the men hunted, but actually the men gathered just as much. In the picture on the right-hand side, the man has come back from a 3-day journey. And he's carried that food without eating it at all because he's sharing. And so the feature of hominins that goes back to life history theory is that we were totally dependent on each other. Now, here they are walking across this Kalahari desert. And if that mother was to leave her baby underneath that bush for half an hour, what would she find when she came back? Nothing. Leopards and hyenas ate the people. And we know this from 2 million years of history in our fossil records, also from South Africa. And so the hunter-gatherer was totally dependent on others. And the child even more so, starting on total dependence with mother and eventually total dependence in the family. And the father was frequently and closely involved in care. This 2 million year history of Homo, of our species, human, goes back 2 million years. And it was close and dependent. In the very bottom corner there, you can see a little red circle. For 100 years, we've separated mothers and babies. In contrast to the 30 million years of zero separation that preceded Homo, going back to primates, where there was total zero separation of mothers and infants. A new model with ancient roots. The ancient roots, zero separation. And so when we look at the care pattern, even to this day in extent present hunter-gatherers, the infant is carried, sleeps with the mother, immediately fed, and the father is closely involved, zero separation. So when we describe nurture science now, in modern terms, life history theory is also called evolutionary biology. But now we have a lot more information about the brain that made that behavior. And now we have even more information about the DNA. Now, all of those things are totally dependent on the environment. For species such as primates, 30 million years, the mother is the environment. Nothing an infant can or cannot do makes sense except in the light of mother's body. The mother's body is that environment, and the baby knows that environment from skin to skin contact, where every single sensory input assures the brain that this is the right environment. Doesn't matter if it's a jungle in Papua New Guinea or this concrete jungle where my sister is giving birth in Sweden. It's the same environment, skin-to-skin contact. This is ecological. The ecological definition comes from your Greek, the branch of biology that deals with the relations of organisms to one another. A baby and a mother are two organisms, and their physical surroundings is each other, is mother's body for the baby's brain and the mother's environment in which she is going to be showing reproductive fitness, which is the objective of life. The behavior that we have in our brains is ultimately only about reproductive fitness. And therefore, nothing an infant can or cannot do makes sense except in the light of mother's body. Now, as I sketch this map of nurture science, regulation is what the baby accomplishes on mother, but the mother is also being sensitized to the baby. And in this early contact, breastfeeding is established. An emotional connection is accomplished in the baby downstream, down the time, and in mother becomes attuned to her infant. This leads to resilience. In the mother, it's wellness. This resilience is the platform foundation for future health. In contrast to separation that produces toxic stress. Now, nurture, I've described, and I said it was almost an oxymoron in kind of science. This science we call in neuroscience is well known, and it's the very science that I'm also building on. And here we have the American Academy of Pediatrics, early childhood adversity, toxic stress, and the role of the pediatrician, translating developmental science into lifelong health. This is the health objective. And there is a model which exactly matches the one that I'm going to be describing. But from it, I am taking one single thing. The definition of toxic stress is the absence of the buffering protection of adult support. And so toxic stress, no buffering, no adult support, makes for disconnected parenting and a disordered attachment in the child and vulnerability. Not immediate disease, but vulnerability, propensity, more likelihood for disease across the lifespan. And this health and disease bar at the bottom depicts the population health. Now, nurture brings a key phenomenon into this pattern, and that is the emotional connection. When I was in medical school, emotion wasn't really very important. It came much later. The brain was much more fundamental than dealing with emotion, some superficial thing in the brain. What we now know is that this is not some superficial thing in the brain. Emotions are the root of the brain, the foundation of human and animal emotions has been beautifully described by the work of Jaak Panksepp, one of my icons, my heroes. And he describes that in this profound work, the summary of the central dogma that underlie all processes is that the DNA and the RNA makes protein and a brain, and that brain in turn makes a behavior and everything else. But the environment is critical for all of these transactions. Here we are, the DNA, the brain, and behavior. The environment influences all phases of these transactions. Let me put some more terminology into this schema. The genome contains all our DNA. Epigenes reads the environment and determines what proteins are made suited to that environment. And it creates a brain with connections. It's the connections which makes the brain work. And some of that's called the connectome. And the connectome collects sensory stimuli from the environment and makes the brain wired for that environment, so that the behavior that we have is life history theory, reproductive fitness. And this is embedded in 30 million years of our DNA. We know a lot more about the DNA now, that there's also epigenes. And this is a picture just to say that it's there. I don't have time to explain this in detail. But this is the 30 million years of zero separations. And this is the conic experiment of Meany that demonstrated that the unsafe environment activated the HPA axis. And in his laboratory, he had rats that were lots of time for their babies, and other rats that had little time for their babies. They didn't look after them very much. And the difference in the mother rat was how many cortisol receptors she had activated that made her more caring. And the less cortisol activated receptors, the more cortisol there was left in the bloodstream of the low-licking grooming mother. And they transmitted these epigene settings to their offsprings, who then behaved like their mothers. Now, he could cross-foster. He could take a baby from a high grooming mother, and in 7 days of no much time care, low licking and grooming care, that baby that did have cortisol receptors and therefore would have been a healthy mother rat and becomes an unhealthy mother rat because it's got high cortisol in the system. The receptors have been switched off in 7 days. Early stress alters gene expression with health impact across the lifespan. He could do the reverse. He could take a baby rat from a low-licking grooming, and by giving it to a high-licking grooming mother, in 7 days, he could switch on the cortisol receptors, and now we've got a healthy mother, healthy behaving rat. And so early care at birth matters because of toxic stress, because of the buffering protection of adult support. The environment is providing buffering protection of adult support to the baby, which in turn influences how the brain is wired. Again, our time is short. And the key thing is about sensory stimulation that fires and wires connections from the sensory organs to the cortex. Now, it's during sleep cycling, 1 hour sleep cycling, that these connections are consolidated and that memory formation pathways are created in circuits. For us as adults, we remember things because we have these circuits. For the baby, the circuits are being created during sleep. And therefore, sleep is absolutely fundamental. Protecting the sleep-wake cycle is what makes brain wiring takes place. And so, neural circuits that are processing basic information are wired earlier. And then on top of those, we can build more complex layers. Higher circuits build on lower circuits, and skill development is much more difficult if the lower level circuit wasn't wired properly. Therefore, it does matter how things were started. The beginning matters most because that's the foundation on which we build. I mentioned pathways, circuits. We connect those into networks, and the sum of those we see in what's called tractography. And here is a beautifully healthy brain with solid foundation extending into the cortical functions. Now, I spoke about rats, and some people say, well, that doesn't apply to humans. This quote, "It's a serious mistake to assume that the principles derived from careful animal studies do not apply to human infants. The risk of suppression or disruption of needed neural processes is very significant and potentially lasts a lifetime." The needed neural processes is the key phrase that summarizes how the brain develops in the environment of, for the newborn, skin-to-skin contact. Now, in skin-to-skin contact, we see a very stereotype behavior. We didn't recognize this 50 years ago. Anne-Marie Wiedström sadly died just this year, and she described this behavior. And suddenly we realize this behavior that the baby looks very random takes an hour, but it's highly conserved. It's in the DNA. It's in the epigenes. It's expressing a brain behavior ready at birth, which makes hormones, that makes neurotransmitters, that makes bodily movements that we see as behavior. Now, we can see this behavior, but we didn't realize how deeply embedded it was in the fundamental fabric of the cell's DNA, of the brain's organ behavior, and of the body as a whole. Here are the 9 steps that she's described, and each of those is hardwired and critical to make circuits in the brain that makes behavior. Highly conserved neuroendocrine behavior. And so Nurture is about not just knowing that these things are there, but ensuring that they happen and that they take place and supporting them. Now, at birth, there are two particular pathways that are wiring the brain, the lower layer of the brain. Smell and contact reach pathways, the amygdala. And the amygdala then fires and wires this particular circuit. It consolidates in 4 to 6 weeks, but it starts at birth. And it's called the amygdala prefrontal orbital cortical pathway. Shore spends much of his work describing the importance of this. It is connecting the emotional brain of the emotional brain is the amygdala, and it's connecting to the social brain. We didn't think emotionality and sociality were very critical for a neonate, and yet it's the very pathway that is being cemented at birth. Here, Panksepp describes it as the integration of emotional systems for social affect, and it requires place attachment. It requires temperature, which is what I get when I get to skin contact, and pain. Now, those bottom pathways are oxytocin, dopamine, and cortisol pathways, and they all share the same tract of skin-to-skin contact. Temperature and contact go in the same lateral, medial, spinothalamic tracts. And so it connects to the amygdala, to the frontal lobe. This is an MRI of a preterm infant that has been in an incubator from 30 weeks to 40 weeks. Now, the next slide shows a similar preterm infant that has been with mother through NIDCAP care. The difference, it is the connection between the amygdala and the frontal lobe and the cross-cortical connections. And therefore, maternal presence fires and wires the amygdala to the frontal lobe. Smell, in fact, alone. This is a study from one of my collaborators, Martha Welch, who just produced smell odor cloth from babies starting very early on. Later on, there came some more stimulations. And just the smell alone, the red value on the 124 electrodes shows where there's a difference. Where's the difference? The left, prefrontal orbital cortical cortex. And the left is where the approach is taking place. The right is where avoid is activated. So this is approach orientation. And when skin-to-skin contact comes with approach orientation, the eye is open, and this shows the mirror neurons. And now empathy is being wired by the inner imitations of actions of others. In this photograph, that baby is 12 hours old. It's learning empathy by reading mother's emotions. Emotions. Oxytocin is the hormone I already mentioned, and it enhances interpersonal trust and the ability to infer emotions of others from facial cues in the first day of life, starting in the first hour of life. Here's a study that shows adults on an MRI machine looking at faces and trying to understand the emotions. And in the MRI, you can see that it's the amygdala and the fusiform gyrus. The fusiform gyrus is the face recognizing part of the brain. I've sketched for you a network, circuits that have been connected together by early contact, smell, made approach, made visual connection with mother. We used to think the newborn brain was a blank slate. Quote from Hugo Lagerkrantz. He's given me the slide. And here we have a 6-hour-old baby that's being put into a modern MRI machine. That's very nice. And that baby at rest has 5 resting state networks. Never mind, these are the A, B, C, they're the 5 networks. You and I have them when we go to rest. Now, never mind the names and where they are. Let me tell you where they are. They're exactly on this network that we've just created. The baby's born with these circuits ready to create the network. And in the mother, the same network is being connected. This is infant cues, the suckling on the mother and the voice and the skin-to-skin contact stimulate oxytocin to connect to dopamine. And this makes the baby rewarding. There's no greater reward than the baby. We're connecting the social control center to the reward control center, connection of oxytocin and dopamine. We're going to come back to this network in a moment. And so this early connecting with the baby, inside the baby, also regulates the baby's physical physiology at the same time as it's creating an emotional connection. So here is my colleague Martha Welch and Robert Ludwig. How babies learn. The autonomic nervous system we all know about is a socio-emotional reflex system also. And therefore, the socio-emotional is that it's an ecological enterprise, an activity between mother and baby that cannot be, they're fundamentally the same. We tend to separate them. What happens when we separate them? The absence of the buffering protection of adult support is toxic stress. And this is the work of Hofer, one of Martha Welch's also colleagues, a wonderful man. And he shows that the interactions between mother and baby are oxytocin-based. And they regulate the physiology. So the interaction is the temperature and the nutrients and the autonomic heart rate. All of those things are regulated by the interaction, mother and baby. Separation separates the interaction. Why does the baby cry? Because it's dysregulated. That dysregulation is quite brief because cortisol is introduced. Cortisol switches off oxytocin. It's antagonistic. Cortisol is my rescue hormone. I use it like that in the emergency room. And therefore, cortisol is producing homeostasis, is restoring regulation in the absence of oxytocin. It's an alternative method of producing homeostasis. But it comes with toxic stress. When it's prolonged, it disrupts brain architecture. And it's the absence of the buffering protection of adult support, which means that cortisol receptors are being switched off to keep the cortisol there. And this disrupts brain architecture. And so the more the disruption interaction between mother and baby that I'm showing from Hofer's research is also in the lower panel showing infant development, maternal care. The closer these are, the more connected they are, the better the interactions, the stronger the health. But when I separate, and here is the basis of these separation studies from an MRI review, a number of things will separate mothers from babies. And those things are disrupting cortisol receptors, increasing the number of methylated receptor sites that switches off cortisol receptors that now doesn't take cortisol out of the system, but rather puts them into the system. And so I'm translating the work of Michael Meany to mother-infant care, and we know it's the same. The ecology in the womb continues outside or not. If it doesn't continue, then we get cortisol increasing in the bloodstream and propensity for disease, vulnerability across the lifespan. And at a brain level, what we then see, I showed you this picture before, cortisol shares stations in the brain. And so oxytocin and cortisol are antagonists. Dopamine is what makes oxytocin win over cortisol. And at birth, the same receptors that I showed you in the resting state networks are the very ones that are activating at this very moment. And so this is called resilience. It comes from the early connecting of this network in the first hour of life and the first day of life. And when we have these connections, oxytocin, dopamine, then we have resilience instead of vulnerability. We have connected reward circuits, and that makes us resilient and helpful, instead of fear circuits that make us vulnerable and prone to disease. Resilience is the capacity to maintain healthy emotional functioning in the aftermath of stressful experiences. Resilience. That's the objective of nurture. And separation produces vulnerability. So this is my mind map of nurture science. And health and disease. Here, Panksepp is quite blunt. Separation produces depression, produces propensity to disease across the lifespan. Other work from primates, developing stress neurobiology requires caregiving, requires regulation, requires buffering. Now we need to be able to recognize harm. We need to recognize threat appraisal, neuroception, Porges calls it. Then when we've got this mastered, now caregiving enables us to be resilient and cope with our emotion regulation. And so I've sketched this as briefly as I can as a foundation of what nurture science is. And that nurture science requires this support of zero separation. For 100 years, we've separated babies. For 50 years, we've kept babies in incubators. And we were able to improve their survival. But now we've got their survival good, but cognitive outcomes have not improved in the last 20 years. And so what we see with these cognitive outcomes is a brain deficit matching a pulmonary deficit. It's the same brain-body deficit that correlates perfectly in the reviews. This is from Twillar. And here's a little clue. Let's go back to Stockholm, where I'm based. And this is my partner research, Björn Westrup. And many years ago, he did a study where they were already doing 6 or 8 hours of skin-to-skin contact a day. But now, from birth, they admitted parents to the NICU. Remember, absence of the buffering protection of adult support. Now I've got 24-hour adult support. That's the intervention. And look what it did to severe bronchopulmonary dysplasia. 3 out of 180 compared to 11. There's a profound difference. The parents' 24-hour presence was the difference in the study. And we haven't improved in our modern care. So nurture is the key. Parent presence, constant uninterrupted presence of buffering support. What I've just sketched in this little half hour is summarized in these 2 articles that I commend to you. And I want to continue and do a little bit more from that article. Nurture science developed recently, as I've just sketched to you now. Neuroscience is 50 years old, but we studied brains in isolation. In the ancient roots, they were never isolated. And therefore, I want to dissect nurture science from neuroscience. Neuroscience is focused on the first 1,000 days. We know it. 1,000 days. Actually, at 1,000 days, we know we have a result that we can measure with the safe situation protocol of Ainsworth and Bowlby. Nurture science begins at birth. Actually, it began in the previous 1,000 days. It matters perinatally and conception, but specifically at birth. It's in the first 1,000 seconds, not the first 1,000 days. It's in the first 1,000 seconds, first hour, when transition to extrauterine life is established, when microbiota is accomplished, and when maternal-infant sensitization begins. The first 1,000 minutes is 17 hours, but it's the first day. This is when the regulation is accomplished. It takes 6 hours. And the mother-infant connection takes 20 hours. I can show. Suckling at the breast is established at this time. The first 1,000 minutes is what really matters. And look how beautifully, the first 1,000 hours is 6 weeks. By the end of 6 weeks, you know what's going to happen at the end of 1,000 days. And therefore, nurture science has a different timetable. I mentioned homeostasis accomplished by cortisol. Oxytocin accomplishes homeoresis. It's better than homeostasis because I got the same vital signs perfectly and I'm growing. I'm doing so by oxytocin, not by cortisol that is increasing cortisol receptors. Now, we know as neonatologists that we have unstable babies in the incubators and therefore we say, don't touch them. Any sensation that is unexpected is a threat to that baby and it crashes. But when that sensation, whatever it is, any sensory input that comes from mother is expected, is welcome, is salient, is relevant to the ecological salience. And therefore it says, oh, this is what I need to grow. This is oxytocin, homeoresis versus homeostasis. And therefore, when we've separated that baby, we've made it unstable. And we're preventing it from getting resilience. The key is that there's maternal regulation. We value self-regulation in neuroscience. I don't want that. I want self-regulation. That's neuroscience. I want maternal regulation. That becomes co-regulation. Even philosophers know that we're defined by our relationships as adults. And therefore, maternal regulation is accomplished at the deepest levels and then up to the limbic emotional brain. Now, another key feature with neuroscience is this individual focus, is this reductionist logic, which comes from the randomized controlled trial. And evidence-based medicine is putting together a randomized controlled trial. Now, I want to say how important it is. I am a Cochrane reviewer of a review. And so I know how to do this. And I tell you that it's absolutely necessary, but it's not enough, because we also have to put all the pieces of the puzzle into the real picture. And therefore, it's not sufficient. When we're looking at ecology, we have two individuals, and we have a dependency on a timetable of an environment, many things. Therefore, it's not, we have to be dyadic and family, and we have to look at dynamic systems theory. Another difference, when we look at neuroscience outcomes, we measure Bailey at 2 or 3 years, and cognition is the key thing. And we also want emotional connection, which we measure with Bowlby's attachment. But they're 2 years. And what can we do about them? How? Nurture science, it's connection that we're measuring. And it's measurable even at some weeks. And resilience, we can measure directly in cortisol receptors. And therefore, resilience is the main objective. Emotional connection, attuned interaction, and here is a longer list of some more differences that's from the publication. A lot of differences, but I've just picked out a few, and I'm going to come back to one more. Nurture science provides buffering protection of adult support, and therefore epigenetic optimal development in the right environment, produces a brain wiring that is appropriate to that environment, that leads to reproductive fitness behavior. Nothing an infant can or cannot do makes sense, except in the light of mother's body, and therefore I've derived for you at length, zero separation. Great, you can say, well, I told you I was a Cochrane reviewer, and evidence. Well, I don't expect you to believe this without evidence. Yes, the science is there. The evidence is here also. It's coming. So where is the evidence? I want to share with you briefly, as time allows, our research. Epistos is the research we're doing here in Sweden. Immediate parent-infant skin-to-skin contact. It has this underlying scientific rationale, but that rationale says zero separation is normal. Therefore, separation is an intervention. You think separation is normal, and if I'm going to study skin to skin, I'm coming with a very dangerous intervention that a 28-weeker doesn't tolerate. But my nurture science says, does it tolerate separation? What is the effect of maternal absence? What is the effect of absence of buffering protection of adult support? And when I ask the question in this way, we don't know the answer to all of these questions. It's a clean, open beach. I'm just reversing the question around. Is there harm from separation on all of these different parameters? So this muddy, messy list, let's tidy it up very briefly. Epistos keeps babies in skin-to-skin contact for 6 hours. That's all we're allowed to do ethically. After that, mothers, both groups, can go skin to skin. And these babies are born at 28 weeks to 32 weeks. And we know their gestation, so we randomize them before birth and they have zero separation. And then I've tidied up all those questions in the 2 slides ago, and we have a sequence of events that is derived and a very set pattern of studies that will connect the dots. Notice I've got long-term outcomes and a Bailey at the end of this, but I've connected the dots along the way. We developed this standardized operating procedure in Stockholm, and then we tested it on a unit in Norway. And this is Siren Rettedal and her team, and there's my wife and I doing the training. Then we came back and polished the protocol, and here we're about to launch in Stockholm. These are twins on a grandmother. Why are we doing this in Scandinavia? I want to quickly tell you that we're doing it also in developing low-income countries. And so we think that 500,000 deaths could be prevented if we can lower mortality. The problem is this mortality comes in unstable babies, and these unstable babies at the moment are excluded from kangaroo mother care, from zero separation, because nobody dared to put them skin to skin. And yet now nurture science says we must put them skin to skin. So if we look at mortality, the babies that die in the first 2 days, first week. And those are the ones that are excluded. Yes, so we could save 500,000 babies if we did zero separation, randomized controlled trial. Janet Patterson gave us funding for a planning grant. That became $6 million that was given to the World Health Organization as a sponsor and conductor of the study. Now, we're comparing babies that are unstable, that get immediate skin to skin, except we've got to weigh them and check a few things. So they only start in the first hour. And we compare those to babies that get normal care, which means incubator best care until they are stable. And then after being stable, they get the same care as the intervention group. Notice I've called it the intervention group, but this is because the World Health Organization is conducting the study and according to standards. So here we have 2 studies. It's the identical intervention that is being used in both these studies, because we took a film, and I'm going to show you a small piece of the film from Stockholm, Stavanger, and we taught the method in Tanzania, Malawi, Ghana, Nigeria, and India. This is not a low-income study. This is the state-of-the-art neonatal care that we've exported in the study, 3 continents, to survive in low-income countries and to thrive in high-income countries. The PISTOS study is ongoing, and we've had problems with funding. Nobody believes us. This is dangerous. The immediate kangaroo mother care study intended to recruit 4,000 babies. When we got to 3,000 babies, the Data Safety Monitoring Board stopped the study because of overwhelming benefit from this intervention. The intervention being immediate kangaroo mother care. And the manuscript is under review right now. It's been a long time. But the protocols are published if you want to see the method, exactly what is done in both these studies. Zero separation. And the time is short. So let me share, what does this look like for state-of-the-art care? And I promised you a little video snippet, and that video is from Stavanger, and the baby is 29 plus 4, gestational age, and weighs 1,200 grams. And let's hope that I can make my technology. I have to stop share and come out of this briefly. And so I've got to go back, and I did practice this before. What I've got to do is go to share screen, and now I've got to go to share. I hope this is showing. So there, just at the moment of birth, what you're seeing then is that at 10-15 seconds, the baby is warmed with warmed towels with a cloth. At 20 seconds, CPAP is started. and the father is present, and the baby is warmed with 2 layers of pre-warmed towel and kept dry. The cord has not been cut at this stage. And now you see the mother taking a breath, and she's already starting to focus on her baby. At 45 seconds, auscultation is taking place. And I've switched the sound down, I think, but the baby's making little grunting noises. The condition is good, and therefore we connect a monitor, and the mother is observing her baby, and she's already totally forgetting about everything else. Her baby is with her. This is why she was having birth in the first place. She's exhausted, but she's got to come back and look at her baby. And right now, at 90 seconds, she's totally focused on her baby. And this is the length of the video. The cord has not been cut. So I'm back to my screen here. Can we see this? I think I put my show video panel. I'm back there. So there's a sequence. 0 separation. We've done things quietly. We've done things rapidly. And we haven't disturbed the baby in the process. 0 separation means immediate at birth, even on a 28-weeker, which is our study. 0 separation means also that we provide, these are pictures from the same film snippet and from the training film with the World Health Organization. There's colostrum to be collected, and we give it. And we give care, still neonatal care, in the labor ward. And then we transfer from the labor ward using father. And then in the NICU, the father is doing skin to skin until the mother can come. And so NICU care started in the labor ward. This is what 0 separation also means. Obstetric care continued in the NICU. And therefore, 0 separation means all babies. I derived this from the science of human beings, of Homo sapiens, not of preterm babies. I'm applying it to preterm babies because they need it more. But all babies need it. The smaller they are and the sicker they are, the more they need it. And therefore, at birth and continuous. On the right-hand panel there, you see a mother, 2 days, the same mother, giving phototherapy, and all the total parenteral nutrition and everything else is being given to that baby, skin to skin contact. The incubator's there. I have to x-ray the baby now and then. I have to do all sorts of things. It also means safe technique. That mother, the previous slide, and these mothers are wearing a shirt, and there's a wrapper that keeps the airway open by binding the head tightly to the sternum. This means that the mother must sleep. All your protocol says the mother's not allowed to sleep. My protocol says the mother must sleep. Otherwise, she can't do it continuously. Even the father can't do it continuously because there's a garment that protects the airway. You can put a monitor on as well, but these babies don't need monitors. 0 separation means breastfeeding even of the very smallest and sickest. It means technology. Here you see that in our study, they got CPAP at the very beginning and they continue it afterwards. And therefore, we transport them in this way. Here you also see that we have dedicated research staff that are looking after these babies and mothers in the mother NICU. Here you see an obstetric ward round taking place at the same time as a neonatal ward round on the same bedside on different sides of the bed. This is in Delhi, India. Here we are in Ghana, and here you see that we have to have surrogates. It's not enough to have a mother. In our IKMC study, fathers are a problem just in the culture, not in the home, but in the culture of the hospital. And therefore, we used other mothers, other aunts, relatives, grandmothers. And therefore, we need surrogates. And we need to particularly have those when we have twins or even triplets. Triplets aren't in our study, but here we still have triplets. And therefore, you have to have fathers and mothers involved as a team. And it means bringing in the family into the NICU. In the beginning, again, it's just the father or the other mother. But later, we're going to bring even the other family. This is a picture that's been seen around the Facebook. Yeah, we can keep the family together. We're not afraid. Profound systems change. But this is state-of-the-art care. It means bringing in fathers and family into the operating room in the first picture. It means bringing the family into the recovery room and into the NICU, state-of-the-art care. Dr. Bergman, this is too much. Everybody's in favor of progress. Nobody likes to change. Nobody likes to change. But I want to ask you, what's state-of-the-art care? And so in a few minutes, I want to bring in a totally different perspective, and it's the one of ethics. In ethics, there are 4 principles, and we have to balance all of them. Non-maleficence, first do no harm. But actually, we have to do 2, 3, and 4. 2 is beneficence. It's not enough not to do harm. We must do good. Justice and autonomy. Autonomy means do the absolute for every patient. For my patient. Justice means every patient. And so we have these balancing acts to change. And ethics requires us to change. Because beneficence, our current low birth weight baby care is producing poor outcomes. And we don't know why. We're not actually doing good. Non-maleficence, first do no harm. We believe incubator separation does no harm. Do we know? We actually know that it does do harm. And so for children, justice is all children needs the care. Children do not have autonomy. And the Convention of the Rights of the Child says there's no autonomy for the child. It's not the parent's vicarious autonomy or the doctor that knows best. The child's best interests are of paramount importance in every matter concerning the child. Child do not have autonomy. Child have best interest principle. Paramount. A child's best interests are of paramount importance. The highest net benefit among available options impose obligations on us as adults. There's an obligation to provide the best interests. We have to be advocates for those children in every single thing. And now I'm advocating that nurture science and absence of toxic stress is the best interests. First, do no harm, non-maleficence. But nurture science says beneficence. It's not enough to reduce the risk of harm. Risk reduction comes from my evidence-based medicine, from my definition, RR, how I reduce risk. Nurture science says it's not enough to reduce risk. We must enhance health. The ethical principle of beneficence says that we must ensure regulation and sensitization. We must ensure resilience. Only when we have got resilience have we done the child's best interests. It's not enough to reduce risk. Risk reduction doesn't produce resilience. So our current low birth weight care is producing poor outcomes. Incubators separate. It's a difference between beneficence and non-maleficence. Separation does do harm. Current low birth weight care is poor. We are not doing active good. The active good requires profound systems change. State-of-the-art care requires that we do this. Nobody wants to change. We have to change. And therefore, all 4 principles carry equal weight in decision-making, except for the child. The best interests of the child are paramount. Introducing nurture science, a new model with ancient roots. It's easier to build strong children than to repair broken men. And we have to do zero separation. Nurture science means zero separation. Professor John Lind from Sweden said, the family is born in the labor ward. Thank you.